Protein purification is a lot of fun, but it's a lot of work. And we don't want to go through all that work if the cells didn't even make, or that is express, our protein in the first place. And so often we'll do a quick check with an SDS-PAGE in order to see whether our protein actually got made before we go through that whole process of trying to purify the protein if the protein isn't there. So the way that we can do this is by taking pre-induction and post-induction samples and running an SDS-PAGE to see if we see a band appear in the post-induction sample that wasn't there in the pre-induction sample. So let me back up. We give these cells the instructions for making our protein with the regulatory region and things that is going to make it so that we can induce the overexpression. So we can say on demand, make a lot of this protein. So much of this protein that they might devote like all their energy to making this protein. And they might even make that protein be like more than 50% of the protein in all of the cell. And so what we could do is we could actually see that that protein got made if we can look and see at bands showing us the kind of sizes of all the proteins, which is what we can get with an SDS-PAGE. So instead of going and collecting a whole big culture of cells and breaking them all open and purifying out the protein, we first look at a small sample, we break open a small sample and see if the protein is actually there. And we don't have to go through the whole complicated lysis procedure where we're trying to preserve the proteins if all we're doing is an SDS-PAGE. So an SDS-PAGE is basically a way that we can use gel electrophoresis to separate proteins by size and then look and see bands that show us basically where the different proteins are in size-wise and how much of different size proteins there are. But we don't know the identity, but that's okay because we should see a band that's there after we induce it that wasn't there before we induced it. And so typically what we do is we induce with IPTG, or like I like to do is auto-induction. And so what I do is I take a 1 ml sample of the starter culture, and that'll be my pre-induction sample, and then I take a 1 ml sample of when I go to harvest it, and that'll be my post-induction sample. If you're doing IPTG induction, then you can take a pre-induction sample right before you add the IPTG. Go ahead and spin those down in the microcentrifuge for 10 minutes, max speed. Remove all the media. You can stick them in the freezer till you're ready. Resuspend them in 300 microliters of your 1X SDS or LDS loading buffer. And then heat them up at 95 degrees Celsius for 10 minutes. Spin them down again for 5 minutes. To remove all the gunk, you want to basically just take the supernatant now, put it into a new tube, load 10 microliters of that on your gel next to a ladder. Nice big band in the post-induction that wasn't there in the pre-induction, which means that now it's worth our time and energy